I'm fine with interruptions at any time. It usually doesn't bother me. So I'm going to talk a little bit about multi-omic studies to illustrate common human diseases. I'm going to talk about that through two studies, ECHO, which is just on the cusp of being able to do some really novel and interesting multi-omic studies, and REDS 3, which is a study that's about 5 or 6 years ahead of ECHO, which shows kind of the power of multi-omic studies. So, like I said, my name is Grier Page. I'm Senior Director of Statistical Genetics at RTI. RTI is a not-for-profit research organization. We're kind of similar to most of the formal labs. I'm entirely NIH supported. RTI is 85% US government grant and contract supported, so we're just a not-for-profit research organization, not a formal pharmaceutical company or anything like that. So mostly I just do my own research. So like I said, I'm going to talk a little bit about ECHO, then REDS. I'm going to talk about a couple of studies within REDS, both the RBC-Omics, which stands for red blood cell omics, talk about some metabolomics, GWA effects, some gender sex effects, as well as get into the Brazilian sickle cell cohort, where we've done a lot of interesting genetics and starting to get into some interesting multi-omic studies as well. So ECHO is just entering its 5th year. It's a large multi-center study that is actually building a cohort of cohorts. There are within ECHO, the goal or the official mission is to enhance the health of children for generations to come. But really the goal of ECHO is to try to address hard or complex human or disease or complex questions of childhood that haven't been amenable to smaller studies. So the goal was to bring together very large sample sizes in order to address complex questions. The complex questions that have been posed are pre, peri, and postnatal health, lung development, allergy, asthma, as well as neurodevelopment and positive predictive health and obesity. Those are ones where there have been numerous smaller studies, but have had variable success. And so the goal of ECHO was to pull together a cohort of cohorts. So the goal of ECHO is to build a cohort of about 50,000 children, and they call them their families, but mostly we've just recruited mothers with very few fathers so far. The goal is to follow children from as close to conception through at least age 5. But what they've done, or what ECHO was set up to do, was that they've adapted and pulled in a number of existing cohorts. And so there's a number of cohorts where the kids are actually now almost 20, and many kids that are new. So ECHO was pulling together this diverse database from studies that exist, as well as doing and conducting longitudinal studies. It is a large, complex study. On here, there's, I think, 62 awards, including 35 cohort sites, data coordinating centers, clinical coordinating centers. There are IDEA state clinical trial centers, as well as genomic cores, metabolomics cores, things like that. It's a large Office of Directors study. ECHO is going to be blessed by quite extensive data. There is they're leveraging extensive existing data. We're generating new data. There's approximately 250 different forms and questionnaires that ECHO participants are going to be filling out during their childhood. Extensive lab data is being collected, EMR data is being generated. There's a strong collaboration for, they call it the HERE core, but that really is doing metal and metabolomics and data generation. We're generating genomic and epigenomic data on all of the subjects and all the children and their mothers as well. We don't yet have funding for epigenetics, but we're hoping to do some interesting epigenetic studies because about 15 of the cohorts already have epigenetic data, and we're pulling it together to do interesting integromic analyses. Microbiome data will be available, as well as quite extensive clinical trial data from the IDEA states and a lot of administrative data. And by that, I mean, we're set up to do an extensive amount of GIS data to make an assessment of exposures. We are collecting people's, both mothers and their children's place of birth and place of residence so that they can if the consent allows, be tracked and their exposures monitored throughout their entire life. As I said, ECHO is entering its 5th year, and so it's really just starting to bear fruit. So ECHO the goal of ECHO was to get a very racially and ethnically diverse cohort. We are currently approaching the recruitment goals, but what we have is around 22,000 or almost 23,000 Hispanic, 40,000 white, 11,000 Black, around 3,000 Asian, and various Native American and multiracial people. This numbers are a combination of both the children and the mothers, so it should ultimately be over 100,000 once we get to the final sample size, but we're beginning to approach the final sample size. One of the other advantages of ECHO is that an extensive database of samples is being collected, and COVID has kind of put a hindrance on people shipping samples right now, but we are in the process of collecting quite extensive samples from all the ECHO subjects, including blood, cord blood, spot blood, teeth, hair, toenails, placenta, breast milk, so quite a bit of different data from subjects that maybe some of these samples are dedicated to producing DNA for whole genome genotyping, and some are going to be going in for metabolomics, but there is quite a large number of resources being built that investigators can be able to leverage to do interesting future studies. In addition to just the total number of people recruited, the amount of data coming in is quite extensive, and this is just really across tabs of the number of subjects that have data by just the basic sociodemographics by age. So what we have is data on almost 51,000 children at different points throughout their lifespan. So we have some data from children at perinatal all the way up to adolescence. So we actually have very nice longitudinal data within ECHO. My role in ECHO is both as the head of the genetics working group, as well as I'm head of the computational informatics and the development. So a lot of what I'm doing with ECHO is helping to build the data capture systems, the data integrative pipelines in order to enable good complex analyses. And we set up a robust data environment where we have quite extensive data flow from external data, large omic data sets, data from individual cohorts that go in, are stored, put in various databases, and made available for analysis. We set this up in a high security environment, what's called the FSMA moderate, because we do have quite extensive identifiable data. So the subjects are being enrolled and stored, and the data is kept in that environment, and people will come into this environment to analyze it. Because these cohorts take such a long time to assemble, we are under a strong push right now to publish. I think we have a goal of over 70 publications to be written by, I guess, July 1st. It needs to be submitted by July 1st, because our first major data release that's coming out of ECHO is January 1st, or not January 1st, January 31st of this month. So what we are doing right now is finally being able to get interesting data out of ECHO. So what we've been doing at ECHO is establishing the parameters and establishing the data to provide a quite large resource that hopefully various investigators can use and utilize to perform interesting and complex questions. And one of the things that I know Doug wanted me to encourage you to do, and I actually want you to encourage you to do, is look at ECHO. Is there maybe a study that you can use and build off of it? Yes, there's a huge number of investigators involved in ECHO. I think there's over 1,000 people involved in different points, but there is so much data. There's so much potential there. And it is a relatively open resource, especially if you work through collaborations with PIs like Doug and other people like that. This is a resource. I will say that we've only published about 20 papers so far. There's no genomics, no multi-omics yet, but we're really hoping that is something we can start doing in the future. And the reason I'm talking about that, and I'm going to transition to talking about REDS, is because this is about where REDS was about 5 years ago, where we had just completed, it took us 4 years to recruit the subjects, and then began doing some really interesting and novel genomic and multi-omic studies. So REDS is the Recipient Epidemiology of Donors study. REDS is an NHLBI-funded study looking at various aspects of blood transfusion, blood donation. Blood transfusion is the most common protocol, or most common most common treatment that's done in the hospital. And essentially 1 in 7 people in their lives will need blood. However, blood is not a perfect product. It is not something that's manufactured. There's quite a bit of variability between people, and there's quite a bit of variability both in the donor as well as recipient response. So the goal of REDS was to be a multi-center study, actually a study of studies, to investigate various aspects of blood donation, blood transfusion, both within the US and internationally. And within REDS, there was about 10 different studies being run, both internally in the US as well as internationally. And I'm going to talk about 2. The first is the RBC-Omics, which is the red blood cell omics. The goal was to establish their databases of cohorts of blood donors and recipients, and I'll talk more about the genetics as we go along. Then I'm going to talk about the Brazil, one of our cohorts in Brazil, which we established a sickle cell cohort in Brazil as well, and we're going to be able to do a couple of interesting. I'm going to talk about a couple of interesting genetic studies on RED3 Brazil. One of the reasons that we're interested in blood is in the US, a relatively large portion of blood, maybe about 10% of blood, gets discarded. And it has to be discarded both because there's an expiration date of about 42 days. And that's regulated by the FDA. But it turns out the reason that you have to discard blood is because it begins to hemolyze. And when blood has hemolyzed, transfusing it leads to a number of negative outcomes in the recipient. So blood gets discarded, but within the US, blood doesn't get used up often, and blood donations are very uneven within the year. There's quite a bit of donation in the fall and the spring and very little in the summer and the winter. So there's often blood deficits as well as blood excesses within the US. So one of our goals of the REDS3 studies of the RBC-Omics was to understand what are the factors that lead to blood hemolysis, and could we identify the genetic factors that lead to hemolysis and those that don't, so that maybe we could target certain blood donor units to be used earlier and those that could be delayed and stored longer and transfused later. In addition, we're just interested in one of the other aspects of it is there's a number of diseases, including sickle cell disease, hemolytic anemia, spherocytosis, epilocytosis, and a number of other disorders that are characterized by hemolysis as part of their phenotype. And so we're interested in seeing is there a connection between the hemolysis of blood storage as well to the hemolysis that is involved in these other disorders. And this is more or less just going over what I said before about if blood is hemolytic and it goes into a person, it can lead to a number of issues, including sepsis, inflammation, and things like that. And that's why we shouldn't be transfusing overly hemolyzed blood to recipients. And the hemolysis the effects that the hemolysis from hemolytic red blood have similar effects to many of the hemolytic disorders like sickle cell. So what we did in the REDS3 was we actually built a cohort where we recruited blood donors from around the United States. And we purposely went about recruiting a multi-ethnic study. And what we have built is both a cohort of African Americans, Asians, Hispanics, multiracial, non-Hispanic Caucasians, people we call high-intensity donors, which we were very much interested in from a blood donor point of view of, do people that give blood regularly, are they different genetically? We really wanted to see, is there something about people that give blood regularly, are they different? And those are people that we recruited people that have given blood at least 10 times within the past 2 years. So essentially they're giving blood about close to the maximum rate that they can. The maximum rate you can in the US is about 12 times a year. So those people were mostly giving it the full intensity. Then we subjected the blood to a number of hemolytic assays, storage assay, osmotic assays, oxidative assays, just to try to establish just how robust the cells is and to establish a phenotype for the blood. Then the cohort underwent quite a bit of analyses. We did, and I'll talk about it later, we tracked the blood. Like when we collected the samples from the recipients, what we did is donors would give blood, we would pull off just a few tubes of blood, and then the unit would be passed on to be donated. And what we did is we built a donation where we actually tracked the blood into the recipients. And because many of the people, like the super donors, give blood repeatedly, we actually are able to track and understand and track the other donations that those people made to the recipients. And at the end, I'll talk about some of the quite exciting results we're getting out of donor recipient genetic analyses. We generated metabolomic data on about, in a time series, but about 200 different subjects. We conducted GWA analyses as well as quite a bit of clinical data and evaluation. We have a pretty extensive data on pica and restless legs and a few other disorders that I'm not going to talk about, but we're also interested, those are also blood-related disorders and are ferritin-related disorders. All of our REDS3 subjects underwent whole genome genotyping data, and this is really just the QC, where we just went through the SNPs, did the QC, and ended up with a pretty decent sample size of 12,658 people. We also were interested in, in addition to generating the genomic data, we generated metabolomic data on, like I said, about 200 different people. And one of the things we're interested in is blood, as what was happening as blood begins to age. So we had blood that was stored for 10 days, 21 days, 42 days. We're very much interested in what is changing about the blood, what is leading to hemolysis. So we did some metabolomic data to just identify compounds that are different. And we also were interested in different sites use different solutions to store their blood. And we're interested in what the effect of different storage solutions have on blood storage. And we did a pretty extensive metabolomic data to try to understand where the changes at different time points. And we've published a couple of papers on that about different storage solutions. But I'll get back to and talk about the metabolomic data in a few minutes and when combined with other genetic studies. So one of the things we did for with the genetic data as well as the hemolytic assays is we conducted a genome-wide association study. And we actually this is actually a quite successful genome-wide association study. And unlike many of them, most of the genes made sense. Many of the genes made sense, which was really very nice to us. Many of the genes like spectrin and ankyrin are the first and second most common genes in the red blood cell membrane. Things like aquaporin is a blood group. Hexokinase is active in the blood. Hemoglobin, that's the hemoglobin beta locus. There's also hemoglobin alpha locus. SLC4A1 is band3, which is the fourth most common protein in the red cell membrane. So we saw was actually a very nice GWA study that made a lot of biological sense. It yielded a number of interesting genetic studies. These are not really multi-omic, but I thought they were kind of interesting that when, like ankyrin 1, which is one of our hits, we actually, once we looked at the GWA study, we actually found that there's 2 hits within the gene, and they were independent of one another. We did a conditional GWA analysis, and we actually found that there's one locus here, and this other locus is wholly independent. And what's very interesting is we think we found the causative variant for both. For this one, which is actually the most significant variant is a non-synonymous mutation within the ankyrin 1. That's relatively common in Caucasians, but less in other people. But you can clearly see that this non-synonymous variant, this is the homozygous for the common variant, the heterozygote, and the homozygous for the rare variant. You can see for osmotic hemolysis, you have a very clear phenotype that variant leads to much more hemolysis. This other one is a non-coding gene, but again, it has a very clear phenotype on osmotic hemolysis, but it's in the opposite direction. The minor allele decreases hemolysis, and we started diving into it a little bit more carefully. What we saw was that the variant, this particular RS number, this is from USC Genome Viewer, sits on 2 transcription factor binding sites. So we suspect, and it actually quite impacts both the MAS and the STAT5 transcription factor binding, which are important in red blood cells. And so we think we actually found both coding variants with an ankyrin 1 in this particular gene. And we're in the process of doing some knockout studies on the non-synonymous variant to see if it has similar phenotypes within mouse, and then try to see what else we can do with them. One of the other things that came up is, and one of the advantages of the Reds and also the advantage of ECHO is it's a diverse cohort. So what we were able to do was to do genetic studies not only in Caucasians, which most genetic studies have been done in, but also in many minority populations or non-Caucasian populations, because blood is given more or less, everybody gets blood, everybody gives blood. And it is important to consider the entirety of it, of the diversity of blood, if you're trying to understand what are donor and recipient interactions. A couple of the variants that came up are much more prevalent within certain racial and ethnic groups. For example, one of the genome-wide significant variants turns out to be the Sickle variant. We didn't have anybody that had sickle cell anemia within our subjects, but we actually had a fair number of people that are sickle carriers within the donor pool. And we found that their blood was much less hemolytic than others. We had a number of people that had various alpha thalassemias, and also observed various effects of those variants upon hemolysis. Since then, we've also started doing a number of other studies. Like one of the other variants that came up as genome-wide significant was SLC4A1, which is a band 3, which is the 4th most common red cell membrane gene. And we started doing various murine knockouts, looking at various things, various aspects of knockouts of the, essentially we took a mouse gene and we began to humanize parts of it and did various knockouts and began to see what is the impact of the various knockouts. And we found that the N-terminal end of the band 3 gene seems to regulate red cell metabolism during storage. So we think that there are variants within this particular end of the gene that are regulating many of the red blood cell metabolism. And we've been able to do that through knockout studies. Let's get through that. This is kind of starting to get a little bit into the multi-omics, that it turned out that by comparing our GWA data to certain proteomic studies that had been done on red blood cells, that a number of the genes that we identified, including several novel ones, are actually the most interesting novel one for us right now, which is SEC14L4, was highly heritable from proteomic studies and relatively common. And many of the genes that we had studied and identified in our studies were highly heritable. We also started looking at, once we started moving beyond the GWA analysis, just for looking at the SNP to the effect size, we also were interested in, could we combine the GWA data with other type of data? First, we started combining it with gene expression. So what we did is we began to leverage through the GTEx data with PredictScan to try to see, normally in a GWA study, you go from the genotype to the phenotype of the GWA analysis and you get a SNP analysis. But what PredictScan does is allows you to go from genotype based on pulling from the EQTL data out of, say, GTEx. You can begin to infer the gene expression based on the genotype within a person. And then we were able to start comparing, does the gene expression level correlate with the phenotype? And then is there overlap between the GWA and the gene expression level as well? And what we found was that the genetics seemed to indicate that a number of the genes that we had found are having an effect upon gene expression. So essentially, these are the most significant genes we found out of our predict scan analysis that we found that the SNPs had an effect on gene expression, and that many of those were under our GWA hits, and many of them were our most significant genes. One of the things that's kind of interesting here is that we identified a number of metabolic genes, but most of the structural genes we did not identify. Ankyrin and spectrin are structural genes, which we think maybe don't regulate through gene expression, but have maybe major gain or loss effects, as opposed to gene expression or regulatory effects. We've also did this on some other phenotypes. The first was on osmotic hemolysis. Now we're working on, this is on oxidative hemolysis. And what we can see is we have a very different GWA result. But we actually are seeing a number of the oxidative pathways coming up out of this analysis, G6PD, PX4, glutathione reductase. And now what we've done is G6PD was one of the, G6PD, which is on chromosome X, is one of the ones that came up. And what we can kind of see is we have, because it's on X, there's both women that are heterozygous and homozygous and men that are hemizygous. And what we can see is that there's consistency of the phenotype in both the hemizygous males and actually we have some homozygous females for G6PD A- as well as the heterozygous females. But what we are again see is that there's just these variants are, this particular variant is just primarily prevalent within African Americans, but it will have, maybe it could have an effect on if African Americans receive blood or if they donate blood. But one of the other things we wanted to do within is to understand the G6PD effect a little bit better. Earlier on, I had said that we had undergone, we had subjected about 200 of the REDS3 subjects to metabolomics analysis. What we were able to do is go into those 200 subjects that had had metabolomics analyses, and we identified 7 of those subjects actually were G6PD A- carriers. So we were actually able to go in, look at the specific genotype here, identify 7 people, 6 are African Americans, mostly male, and then begin to understand what is the genetic regulation and what effects are being seen within, what are the differences between those subjects with the G6PD A- variant versus those that don't. And first off, we wanted to see what is the variant or what metabolite, most differentiated those people that had G6PD versus those that didn't. And interestingly, both pyruvate and pyruvate lactate ratio were highly predictive of differentiating between those that carry G6PD and those that have the wild type that are highly significant. And then we've also begun to try to tease apart the metabolic pathways, looking at within the subjects that had been genotyped that we were beginning to see that we had lower glutathione, lower pentose phosphate pathways, and how that began to impact the downstream effects. So we could begin to understand what effect that G6PD might have on people in their normal life. These are people that are healthy enough to give blood, but yet they seem to have altered metabolism, and we can do this by leveraging the data. We started going through this for all the oxidative and osmotic variants to try to, especially upon the metabolic genes, to try to understand what are the effects of specific variants through the metabolite and try to understand how that leads to differences in the metabolism, and ultimately try to integrate them and pull them all together. The other thing we've started doing is, because the REDS3 data, we have, we collected extensive, we collected blood. They subjected them to extensive assays, CBCs, different assays, but also we collected extensive phenotypic data. And so we had smoking data on all our subjects. And we're interested in trying to understand what are the metabolic effects of smoking within a person. So what we've done is we essentially, both from self-report as well as by being able to measure cotinine of the metabolite, be able to identify those people that are smokers and not, and then began to differentiate where the difference is, and be able to identify that the smoking seems to alter energy metabolism, glutathione, and a few other pathways. And we're able to identify the genes that are, and the metabolites that are most, the metabolites that are most correlated with smoking. and begin to understand a little bit about the biology of smoking. We've done this for alcohol. We're doing this for marijuana, heroin. What's very interesting is that you're not supposed to donate blood if you've smoked marijuana or an IV drug user, and we're definitely detecting a number of drug users, and we're within them, just being able to see the metabolic products out of them. But so with this data, which is blood donors, is also going to be able to give us good insights into many other normal mechanisms. And that's something that I'm sure would also come out of ECHO as we begin to study it. Yes, the subjects are collected for child health, but we're also getting extensive exposure data, and we might be able to see and make more complex and different hypotheses than our main ones of ECHO, because smoking and alcohol use was definitely not a main endpoint of REDS, but it's something that we'll be able to drive because we collected the data. I'll skip over polygenic risk score as I'm running out of time. REDS 3, one of the interesting results that came out of REDS 3 on the hemolysis was we found a very marked gender effect. That we found that males and females were quite different on storage, ferritin, osmotic hemolysis, and oxidative hemolysis. And we wanted to understand why do males and females differ, and what is the cause of it. So one of the interesting things, well, one of the things we did is we kind of turned to GWA analysis and did it stratified by gender. And we were interested in seeing, do the genetic effects in the men and the women, are they the same? And somewhat surprisingly to me, we actually found there to be quite a large differences in the GWA profiles between men and women, suggesting that perhaps men and women can be, may have different genetic regulation for hemolysis, or at least some differences in genetic regulation. We're not the first study to report large gender differences by disease in gene regulation, but one of the things we're looking at is trying to replicate this, because while it's been reported a couple of times, nobody has really replicated yet large gender differences in genetics. And what we do have is a number of variants that are genome-wide significant just within specific genders. This is essentially some of the markers that are hugely significant within females, essentially non-significant males and quite significant males, and non-significant in females with very large interactions. For example, this is around band 3, which is one of the genes I talked about. We have a very large, clear signal in females in band 3, and essentially no signal at all in males. Again, suggesting that future genetic analyses should consider gender and look at the differences in genetic regulation by gender. Also, we started looking at, some of the REDS investigators were interested in hormones, and primarily sex hormones, both estrogen as well as testosterone, that some studies I'm not going to talk about. But what we had here was another locus, nearly genome-wide significant in females, essentially empty in males. But what's very interesting is within females, there is a very significant hit, or relatively significant hit, in females before menopause. But after menopause, the effect went away. So potentially there could be regulation by some of the female hormones, genetic regulation of some of the female hormones here. And so we're in the process of following this up with some other studies and some mouse studies, and seeing if we can understand how do estrogens begin to change regulation of some of these genes. But we also began to do some more complex analyses that actually I was kind of surprised that this worked, as we did a primarily GWA analysis. This is on osmotic hemolysis. These are the same locations within the genome. This is the main effect, and we found nothing at all. But when we ran a gene by sex interaction analysis, we found hugely significant results. So the main effects, there was no main effect, but highly significant interaction effect. And what we're seeing here is that within females, there's a rising effect. Within males, there's a falling effect. So there may be different there may be very interactive effects across genomes, and that by focusing just on main effects within GWA analysis, you may be missing results. And some of the, so we're also, there's a lot of other studies going on there that I'm not going to address. One of the points of REDS, or one of the things we were most interested in REDS, was trying to see what is the effect of genetics in recipients. And so what we, as I mentioned earlier, we had taken our blood donors and we were able to track them to the recipients because we were integrated into a number of the blood donation centers. We're working with Blood Center Wisconsin, Blood Center Pacific, American Red Cross, and they have very good data connecting their blood to the components to the recipients. And so we were able to build a large donor recipient database where we could track blood from a donor to the recipient, and we can track longitudinally all the donations that a person has made during a period to the recipients. And in some cases, because some of these are high intensity donors, in some cases there's 35 or 40 blood units that we can track to recipients and look at their effects. This is essentially the same numbers, just rearranged, but within those subjects that we had genetic data on, we could track from the 13,000 blood donors we had, 8,800 of them we could actually link to the recipient, and we had clinical outcome data on. And we have some subjects where we could actually track out to 8, 10, 12 subjects. And we've just recently begun, because this data took many years to clean, unfortunately, but we've recently started seeing that there are, and these are very preliminary data, this is just in one cohort, 1/4 of the REDS3 cohort, where we've been able to start seeing that the donor genetics may have an effect upon the recipient's hemoglobin response. By that I mean when you give a transfusion to a person, the response is measured by the increase in hemoglobin. And typically, if you give somebody a unit of blood or a unit of red blood cells, you expect their hemoglobin to increase about 1 per unit. And what we're finding is that based on the donor genotype, we're finding there to be some substantial differences in the response of the recipient. Like if somebody is a alpha thalassemia donor, we're finding that the recipients have much smaller responses than those that are wild type at alpha globin, and those that are G6PD, either hemizygous or homozygous, are also causing there to be significantly smaller hemoglobin responses. So one of the things we're trying to do now in precision medicine, or what we're calling precision transfusion medicine, is can we begin to start genotyping donors and look at recipients and do optimal donor recipient matching, and then maybe potentially encourage some donors, discourage other donors based on the response, or do better matching of donors and recipients. And this is, we've just printed a new array that we're going to be genotyping essentially a large component of the donors within the US to try to begin to really systematically track the donor to recipient, and really do much more systematic studies to see, are there variants that do predict blood response. Next thing I'm going to talk about is the REDS3 Brazil. I'm going to skip a lot through here because I notice I'm running long. So the REDS3 Brazil is a cohort where we collected sickle cell patients within Brazil. Brazil is within the New World has the second most number of sickle cell cases. They may have somewhere between 50 and 75% of the number of sickle cell cases as compared to the United States, because there's a very large African ancestry that came into Brazil that brought in a number of sickle variants. Brazil has a very interesting blood system in that because one of the main treatments for sickle cell disease is blood transfusion, that many sickle cell patients are essentially treated at blood donation centers. So that's the reason we began to build this sickle cell cohort because the sickle cell patients were being treated at the same place that we were beginning to do genetic studies of blood in Brazil. And it also makes it very easy to do follow-up with the sickle cell patients because they've been coming to these hospitals for many years, and there's quite extensive longitudinal data. So we were able to build a cohort of about 2,795 subjects with sickle cell disease, and this is the largest sickle cell cohort in the world right now. We were able to get all of them whole genome sequenced as part of TopMed. So we're in the process of using that whole genome sequencing data on the Brazilian sickle cell patients to understand, because sickle cell is a monogenic disorder, we know why they have sickle cell, but sickle cell has quite variable expressivity and expression. And so we're interested in trying to understand, why do some people get stroke? Why do other people not get stroke? Why do some people have acute chest and repeated hospitalization, quite severe effects? So we're trying to look at many of the different genetics of the sequelae of sickle cell. I've done a lot of population genetics in the Brazil that I may just skip around and get to the biology. One of the sickle cell disease, I said, is a major disorder, monogenic disorder, but one of the major known modifiers of sickle cell disease is retention of fetal hemoglobin. The beta globin locus is actually a multi-gene cluster, and developmentally, humans begin expressing first an embryonic globin, then a fetal globin, then an adult beta globin. The sickle variant is in the adult beta globin, and that causes there to be the beta globin to be defective. But sometimes the fetal hemoglobin can be retained, and that will compensate for the lack of for the defective beta globin variants. So the best predictor of sickle cell outcome is the percentage of fetal hemoglobin retained in childhood. So we're interested in trying to understand, and that's been a known phenotype for quite some time, and we're interested in understanding the genetics of fetal hemoglobin regulation, as well as trying to determine if the genetic regulation, one of the other questions we're trying to get at is the genetics of sickle cell, or the genetics of sickle cell sequelae in Brazil, the same as the US? Because there was some worry that, I apologize for jumping like this. The slave trade, which brought people of African ancestry to Brazil, came from very different parts of Brazil than did the slave trade into North America, with the variants that came into North America, or the slave trade mostly came out of the English colonies in Africa around Gambia, Sierra Leone, Nigeria, Ghana. And most of the Africans brought over during the slave trade to Brazil came from the Portuguese colonies, kind of northern Mozambique, Central African Republic, a little bit of Mozambique, things like that. And so there was some worry that the sickle disease was different in South America than North America, because there are different sickle variants within Brazil from where the Portuguese colonies are versus where kind of the English colonies were. So one of the things we're interested in seeing is the genetic regulation of fetal hemoglobin the same between North American, African American sickle cell patients and Brazilian sickle cell patients. And it was known that through previous studies, such as OMG, Walk Fast, Push, that there were 3 known loci that had been known to regulate hemoglobin beta, BCL11, MIB, and the beta globin cluster. And so we're interested in seeing could we replicate those findings in the Brazil sickle cell. And so we conducted a genome-wide association study on the percent hemoglobin within the Brazil sickle cell cohort, and we actually replicated all 3 known variants within North American African Americans, as well as identified 2 novel loci within the Brazil sickle cell cohort that regulate hemoglobin F. One advantage that, well, I said hemoglobin F is the most important regulator of sickle cell severity. One drug, which is called hydroxyurea, which is also a chemotherapeutic agent, is known to increase fetal hemoglobin levels. And so it's often given to sickle cell patients in order to increase the fetal hemoglobin and also to reduce the symptoms. And because it's a pretty powerful drug, not everybody can get therapeutic doses. So there's also some interest in trying to understand the genetics of the toxicity of hydroxyurea. But one of the things within the Brazil cohort, we had enough subjects that we could do GWA analysis both on and off hydroxyurea. And oh, there it is. Within the Brazil sickle cell, we had 1,700 subjects. We had some without hydroxyurea treatment and some on hydroxyurea treatment. So we were able to do a GWA analysis for both of these. And what's interesting is the Red 3 Brazil is a longitudinal study. So for 168 of the people during the longitudinal study, they actually changed hydroxyurea status. So we're actually able to do some genetic analyses within people to look at what is the genetic regulation not on hydroxyurea versus on hydroxyurea. And what we found was that the known variants within that had been previously known from North American African Americans, the BCL11A, MIB, and hemoglobin beta, held up very consistently in those with no hydroxyurea, but those on hydroxyurea seem to have a very different genetic regulation. And what we're finding is that hydroxyurea, like in those without hydroxyurea, these are the known variants, are highly significant. This is the effect size of an allele. What we're finding is that the hydroxyurea is reducing the effect size in this, of that variant on hemoglobin F level. And so that's why they're becoming non-significant. But what we're also seeing is that the hydroxyurea is increasing the effect size from almost doubling from not on hydroxyurea to on hydroxyurea. Why that becomes significant. So what we're finding is that the hydroxyurea is quite extensively changing the genetic regulation of fetal hemoglobin within sickle cell patients. And we've developed polygenic risk scores to predict hydroxyurea and both predict baseline fetal hemoglobin response, so that if you go into a newborn that has sickle cell, you'll be able to predict how well what their fetal hemoglobin will be above H5, and thus have an idea about the symptoms, but also we can predict if they're going to respond to hydroxyurea treatment or not. So this is definitely not my entire work that we have. I have quite a large, extensive collaborative groups, both with REDS and with metabolomics groups and sickle groups and things like that. So what my goal here was to say that about 5 years ago, REDS, we spent 4 years collecting the data and really hadn't done any analysis, but we had potential. There was samples, there's data, and that now there are at least 7 or 8 different R1s funded off of this REDS data. There's what's now called the REDS 4, which is a successor grant to that, continuing the studies. And that one of the things that is now what is similar in ECHO is there is a great potential there. The data is almost here. The samples are there. There will be the potential to do really many very interesting multi-omic studies within ECHO over the next few years. And I encourage you to get involved if you can. And so thank you. And I'm happy to answer any questions you have.
Okay, thank you, Grier. That was a fascinating talk, and it makes me a little bit more hopeful about ECHO finally coming out with something.
Well, it's one of the things about like REDS was, like ECHO was initially funded in 2016, REDS was initially funded in 2010. Mm-hmm. We really didn't have the data until late 2016, late 2017.
I imagine REDS isn't funded as highly as echo too, right?
No, not even close. No, it has probably a fifth the funding, a sixth the funding of echo, if not less than that. Okay. It had a lot of funding because collecting 14,000 blood donors is a lot of money, but it didn't. Echo is much larger.
So we have about 5 or so minutes for questions. I've written down several questions, but I want to give everyone else a chance before I dominate. Does anybody have any questions? Please unmute yourself.
Probably easy one. Thanks. That was just a great talk, and like Doug said, it is very encouraging for Echo. We're coming into some exciting times. I noticed early on when you put up your participant flow chart for REDS, you noted that one of the things you were assessing was plasma ferritin, and that you gave some results on that. And plasma ferritin, of course, can be used as a marker for iron deficiency, and if it's high, for inflammation. And I just wondered if you could say a few words about what your rationale was for using that.
Well, back to it. Well, the reason we measured ferritin was, I guess, I wasn't involved in decision making on why ferritin was chosen, other than there was previous studies in REDS 2 had noted that there was extensive depletion of ferritin from donation. Can y'all see this, or can?
Yep.
That blood donation causes there to be extensive depletion of ferritin, and there are other studies going on trying to understand what are the clinical consequences of this. Because one of the things that we're doing is that ferritin is also associated with both pica and restless legs, and there's other studies that are going on looking at, as the ferritin depletes, do people and blood donation depletes people, are they becoming more susceptible to pica and restless legs? Pica is the disease where people eat non-nutritive substances like chalk and clay, or in America, mostly ice. And restless legs is the tendency of people to jump, which is quite associated with ferritin levels as well. And so it was collected in order to both compare the results here versus hemoglobin, because like all these are blood donors, so they all have sufficient hemoglobin to donate blood, but yet the reserve iron stores are going down. So there's interest in trying to understand why hemoglobin is being maintained while all the reserve iron stores were going down as well. And there's a bunch of other studies on there.
Cool, thank you.
So one of my questions, Grier, is in your GWAS effects, you showed these really strong sex-dependent effects. Is that more often than not, or is that just a small fraction? I didn't get a feeling for that. It seems like based on this graph here, it seems like almost all of them are sex-specific effects. Or is it, what would you say?
There are several that are, like some of the hits are actually going in, it's not as obvious, but there's ones going in both directions, like here, and most of them
major hits, though. I guess the way I meant to say the major hits seem to be sex specific.
What we're seeing is that what's interesting is that there are genome-wide hits that were genome-wide in the entire data set, and then once you pull it out, they're only a male or only female contribution, only male contribution. And there's a bunch in here where there's about equal contribution of male and female through here. But
do you think this is entirely hormonal, or what else could be doing that? Y chromosome, 2X chromosome?
It could be hormonal, it could be developmental. One of the things we're doing now is both trying to make sure that this isn't just randomness. And so we're in the process of doing other GWAS studies, like we have CBC data on all these subjects, and we're looking at are there gender effects of CBCs, and just trying to get a null distribution of what are male and female effects. And then what we're doing is also just randomly dividing data and to try to get null distributions of once you split it, to make sure that we're actually seeing real signal. These are like these on 17 and are genome-wide significant. So we've anomaly done an adjustment for multiple testing, but it still worries us a little bit that we're not seeing real effects, that we need to deal with some of the statistical issues. And that's one that we're dealing with. We have the people I work with, named Fong Fong, has grant to try to understand better the gender effects and to see both how prevalent are gender-specific genetic regulation, and also to show that both it is replicable, because with CBCs, if we see them, there's lots of data we can leverage there. One of the problems we've had publishing our paper, which is that we don't have good replication for our hemolytic phenotypes because nobody else has really looked at hemolysis before.
Have you like measured estrogen levels or looked at the time in the estrous cycle that the blood was donated and that sort of thing to control estrogen levels?
We have that data. We have it from the metabolomic data. So yes, we also have if they're on birth control, if they're on Premarin, things like that, because a large portion of most blood donors are mostly older. They're more than half are over 45. And so we actually, a large portion of our women are postmenopausal. It's about half premenopausal, about 2/3 postmenopausal, 1/3 premenopausal. And then of those that are premenopausal, there's a huge group that are high school students and college students. It's a weird distribution. We have a weird age distribution in this data. There's a big peak in high school and college. It kind of goes down and then peaks again, something around age 50. It's race dependent too. But
okay. Well, another question I had was, so you mentioned like your Brazilian cohort, you're doing whole genome sequencing, and ECHO, I think they decided to do like 1 million SNP arrays. And that seemed kind of is there a huge effect? I mean, it doesn't really, it's unlikely you're going to get causative SNPs. How important is it to do whole genome versus a smaller SNP array type thing?
I didn't put it in here, but within the REDS3 Brazil, we have both whole genome data, and we have the same array we ran on the RBC-Omics, and we started doing comparative analyses. And truthfully, I'm finding the whole genome analysis to be not that much more useful than
that's not surprising to me, because you're not typically mapping down to causative alleles. You just need regions of the genome is good enough.
Because once you do a minor allele frequency filter, like we usually do our analysis at variance at 1% minor allele frequency, the number of markers you get out of whole genome sequencing versus array that are high quality is about 5% different. And in some cases, and we have in some places we have more markers on array, in other places we have more markers on whole genome. And the whole genome data is not, there seems to be holes in it or issues with it. Like in the multi-gene regions, the whole genome data doesn't work well.
Right.
Like we've actually done the hemoglobin F analysis in both array data and whole genome data. And hemoglobin beta is one of the regions that the whole genome doesn't work. And so that effect is gone in the whole genome analysis.
Right. It's kind of similar to like the genetic testing of newborns, where they had some RFA a few years ago that we applied for and didn't get, where whole genome versus, the 50 gold standard tests that they do for different genetic diseases. And whole genome wasn't nearly as good as the standard test. So there's some reason for not doing whole genome. So whole genome is a lot more expensive and it's not as good.
Yeah, like so that when we've been running them, we've been having some issues comparing them. But the place where we've had the most utility in it is in our some of our population genetics. This is we're interested in the origin of the sickle variant. This is what I skipped over, was that because we have in TopMed we have like 4,000 sequenced sickle cell patients. We're able to get really detailed sequencing data and really try to resolve it, because the sickle, I talked about the haplotypes. There was, for many years, there were thought to be many different, the sickle variant was thought to have risen multiple times, and based on RFLP haplotypes that were quite large over the region. And we were interested, and then a paper came out, I guess now 2 years ago, from Daniel Schreiner, saying that there really is only one variant, a single origin of sickle variant. And so they did it off the basis of 129 haplotypes. And so we thought we have 6,000 people. Let's begin diving into it. And one of the things off a whole genome is you can get really, really precise haplotypes. This is essentially, the blue is the minor variant and the yellow is the major variant based off of European populations. But this is essentially all the variants within 2 kilobases of the sickle cell allele. And what you can see is there's a ton of variants. And once you boil it down, what we found was that there, at least within 500 kilobase, not 500 kilobases, 500 bases of the sickle variant, everybody has the same haplotype. But once you get this, like I said, this is the sickle variant here, this blue line, and the C is here. But this variant here, where there's an obligate recombination, is 500 bases out. And here, this is, this is 560 bases out. So there's obligate recombinations at 560 bases out. So what we're trying to do right now is mathematically determine if it's more likely that there were 2 recombinational events within 500 kilobases ancestrally, or if there's a single versus multiple events. And I'm coming to the conclusion that there probably was a single origin of sickle variant, and there's been a lot of recombination there. But the whole genome is awesome for doing really, really detailed population genetics and dissecting variation and things like that. And what we've done, we're able to do, because in the sickle cell, they're all sequenced. Once we find variants of the GWA study, we can dive into the data and really find out, look at the minor alleles and things like that. So the actual discovery, the whole genome doesn't help with, but some of the follow-up, the whole genome does. If you can connect, you can look in the sequencing data and identify the specific variants that they have, because often when you're doing a GWA study, you're typing relatively frequent alleles that are non-coding, and that you really are usually interested in coding variants, and that by going into the sequencing data, you can determine if people carry relatively rare variants.
Okay, well, thank you. It's 5 minutes after the hour. Some people have left for other meetings.
Okay.
Thank you very much, Grier, and
Thank you.
We'll keep in touch and hopefully do some genetic studies with ECHO in the future.
The call is coming out on Tuesday for all the genetic data to come in.
Great.
Yep.
Okay, thank you very much.
Thank you.
See you all later.